See, the top one is the ectomycorrhizal, the characteristic feature of ectomycorrhizae in angiosperms, and the bottom one is the characteristic feature of conifers. So in conifers, it enters within the cortical region, but it will be intercellular. It occupies intercellular space. It will not penetrate within the cells. So that is the main difference between ectomycorrhizae and endomycorrhizae. Okay, and you can see here in angiosperms, it is restricted to only epidermal cells. So this is the transverse section of the same thing. So this is angiosperm, and this is the other half of the figure is this angiosperm, and towards right it is conifers. So this is ectendomycorrhizae, wherein you can see the space in between the cellular intercellular space also is occupied, and the part of the mycelium enters within the cell. So that is the main difference between ectomycorrhizae, ectendomycorrhizae, and endomycorrhizae. You can see here space is occupied in between cells, and it also entered within the cell mycelia. Again, this is also an ectendomycorrhizae. This is how it looks. So this is a pictorial diagram. Actually, it is a 3D structure. You can see here. Coming to this arbuscular mycorrhizae, you can see the arbuscular mycorrhizae colonizes through this the spores which are there in the soil, or through the neighboring root debris which has this mycorrhizal mycelium, and whenever it finds a host, suitable host, it enters and colonizes, and it occupies and enters within the cell. So there are two types of mycorrhizae they have reported. One is Aram type, and another one is Paris type. And in Aram type, the only difference is Aram type and Paris type is in Paris type, we can see here coiling of mycelia takes place within the cell. And from this coiling, you can see the arbuscules emerge. Whereas in case of Aram type, the coiling is not that much. So arbuscules is a finger-like projection or a tree-like projection in case of Aram type. That is the main. These are the main two types you can find whenever you screen plants for the presence of mycorrhizae. So this is a required mycorrhizae. This is restricted to the required plants which are restricted, which are known to grow in temperate zone. So these are also endomycorrhizae in nature. So it penetrates, and until and unless the plant forms association with this type of plants, mycorrhizal association with this plant, the plant cannot complete its life cycle. So because the time available for the completion of life cycle is very less, and acquisition of nutrients, these mycorrhizae help in acquisition of nutrients. That is why it has to form association. And this is another one type of mycorrhizal association found in arbutoid plants. This is also endomycorrhizal in nature. And this is monotropide. This is, you can see here, ectendomycorrhizae. This is, you can see, it forms a peg-like appearance, and which enters into the fungal peg. It is called as fungal peg, and which enters within the cell, and it covers the entire cell surface, you can see. So this is another one type, orchid mycorrhizae, and almost all orchids actually, as of now reported, they are known to form mycorrhizae as soon as they produce this corm, you can say. And how exactly the mycorrhizae enters, it is still a puzzle. So if you are interested, you can take up this research, and you can find how exactly the mycorrhizae enters the seeds, because these are the recalcitrant seeds. So during in natural condition, they lose viability very soon. So in order to establish its the corm, the mycorrhiza plays a very important role, and as the plant grows old, the mycorrhizal mycelia will degenerate, and that is utilized by the host plant actually. Okay. So coming to this arbuscular mycorrhizae, I am going to restrict arbuscular mycorrhizae. So arbuscular mycorrhizae, there is just to begin with, innovation, if spread globally, becomes adopted to multitude of diverse application and persists over eons of time, and it can be considered as a great success. So this particular association is considered to be a great success during the course of evolution, which helped the plants, particularly land plants, in establishing and sustaining over a period of time actually. So whatever, as of now, I think whichever the plant you just take and screen for mycorrhizae, definitely you will find a mycorrhizal association. There are few exceptions. So it is known these plants growing under conditions where they face deficiency, particularly with the immobile elements such as phosphorus, copper, zinc, and other are considered to be the special significance, and as their availability towards the plants are limited to the soil, and because the mycorrhizae has access to the extra root area, you can say. So that will help the plant to reach wherever the resources are available, particularly with that of phosphorus, and it helps the plant in acquisition of these kind of elements. So as of now, 80 to 90% of arbuscular mycorrhizal fungi are considered to be forming association with the angiosperm plants. 80 to 90% of the angiosperm plants are considered to be mycorrhizal in nature, and they are obligate biotrophs, fall within the Glomeromycotina subfamily, subphylum, and of the Mycorrhizomycota. And Glomeromycota represents almost 1,000 species, and yet only 300 have been described morphologically, because it is considered to be very difficult to identify morphologically based on the spore structure and the content within the spores, the hyphal attachment, color of the spores. So these are the characteristic features, and based on these morphological structures, as of now, 300 spores have been described. And this is relatively a small group of obligate symbiosis, and with diverse range of plants spanning almost 2,000 species, generally have been with low host specificity. That means these mycorrhizae, they doesn't have any specific host actually. So wherever there is availability of the root, you can see, and the root exudates, if they sense or if they come across, they tend to form association. There is no host specificity, you can say. Okay. And AMF symbiosis appear to have emerged around the same time when the bryophytes have been evolved. Actually, this we have discussed. Okay, so as I mentioned in one of my previous slides, early fossil records confirm this hypothesis, as fossils from almost approximately 407 million years show structures that appear to be like arbuscules. And AMF symbiosis is formed when the fungus penetrates the plant cell wall, but it will not damage the plasma membrane, and forms an integrate branching, which are called arbuscules like structures within the cells. So this is the homeophyton, actually the earliest plant, wherein they have found two different type of structures. One similar to that of vesicular and arbuscular type, and another one is similar to type of this D, E, and F. You can see the mycelia similar to that of Mycorrhizae. Okay. And here also you can see the extant land plants, particularly the rainy chert fossil plants, similar to that of the extant land plants. You can see the mycorrhizae arbuscule-like structures they have formed. So this is the evidence. They want to say that yes, the earliest land plants have also had mycorrhizal association. Okay. Coming to this mechanism of plant root colonization by arbuscular mycorrhizae. So I think this we can, okay. So when the spore germinates and starts searching the host, whenever it come across with the exudate, particularly the exudate which is considered to be attracting mycorrhizal spore is the strigolactone, which is a hormone released by plants, like that of flavonoids released by plants to attract rhizobium. Here, strigolactone is considered to be one of the most important plant hormone which attracts the spores and mycelium. And once the spore come in contact with the root, it forms a hypopodium. And once it forms a hypopodium, a signal transduction takes place within the plant's epidermal cell, and it penetrates the mycelia, enters within the host cell. And once it enters and reaches the cortical cell, it forms arbuscules. This is the whole concept. Okay. So the difference between ectomycorrhizae and endomycorrhizae is you can see in endomycorrhizae it will be occupying the intercellular space, whereas in case of endomycorrhizae it enters within the cell and without damaging the protoplasm. Okay, with the help of this hypopodium attachment. And the place of nutrient exchange, particularly the symbiotic association in, as you all know, both the partners need some benefit. So mycorrhizae gets carbon source from the host plant, and in turn it supplies the nutrients, particularly phosphorus, and of late I think nitrogen also considered to be one of the important nutrients supplied by this mycorrhizal association, and also some of the micronutrients. So here you can see here the peri-arbuscular membrane. It forms a peri-arbuscular membrane wherein this here it is considered to be the exchange of food material takes place. That is the main region. So you can see it has not damaged the host plasma membrane, and it formed a space in between there that is called as peri-arbuscular membrane. Okay. So there is a clear demarcation zone, and that is the place where nutrient exchange takes place. So biochemical aspects, as I mentioned, the mechanism of AM interaction starts even before the mycelium is physically come in contact with the host root, and particularly in response to the plant root exudates, that too this strigolactone, as I mentioned, the resting spores start germinating, and it branches off continuously towards the reaching towards the root in response to the signal received from the plants. So this AM symbiosis involves a number of signaling pathways which are partially characterized and are identical to those of rhizobium legume symbiosis. You can see most of the steps almost same. You can see LCOs are here, LCOs are here in case of rhizobia, and these LCOs is sensed by the membrane bound receptors. This is lithium and RKS. These are the kinase receptors, and it sends such signal cascades of signal transduction sends within the cell, and that will establish the mycorrhizal association. So as I mentioned, so this particular root exudate, particularly strigolactone, is considered to be the main hormone which is important in recognized by these spores, or you can say the approaching mycelium, which is there within the vicinity of rhizosphere, this root rhizosphere area. Okay. And once this strigolactone is recognized, the mycelium start multiplying towards that, and it forms a hypopodium, and in turn it forms establishment of this mycorrhizal colonization. Okay, this is another one pictorial representation just to mention. So once it this hypopodium comes in contact, the low frequency and high frequency calcium spiking takes place within the host plant, and this makes the way for what you can say growth of this mycelium, and it establishes the arbuscules within the host cell once it enters sufficiently within the inner cortical zone of the root. And to mention few thing about the molecular aspect, as of now, 7 genes have been recognized, wherein they are known to play an important role in mycorrhizal association within the plant. So they are the receptors like kinases, okay? The protein receptors, the kinases, and they are similar to that of rhizobial recognizing kinases. And in mycorrhizae, chitin oligosaccharides and lipochitin oligosaccharides, these two are the two short chitin-derived compounds which are considered to be the effector proteins which play an important role in AMF symbiotic signaling, where the plant recognizes and allows this particular AMF to form association, because within the rhizosporic zone there will be a lot of organisms, as you all know. So why only mycorrhizae forms could able to establish association? Because of the signal recognized by the plants, you can say. Okay. So once the plant reception of LCO activates, once the plant receives the signal, this LCO actually activates this calcium oscillation, as I mentioned, and within the plant nucleus through well-conserved set of genes, and this forms a common symbiosis signaling pathway, as here you can see here. Once it comes in contact, so it sends signals, that is high frequency calcium spiking and low frequency calcium spiking takes place. So that itself will be the main reason for calcium, this mycorrhizal association. And another one is like NOD factor in case of rhizobium. Here in case of this arbuscular mycorrhizae, which releases a MIC factor. Okay, and that is considered to be responsible for this host root colonization in the presence of strigolactone. So, as you might be knowing, this NOD factor plays a very important role in nodule formation. So, in case of mycorrhizal association establishments, also MIC factor plays a very important role. MIC factor, and that too in response to strigolactone. Okay. So you can see here the pictorial representation. Once the AMF receives signal in terms of as stygolactone leaches out from the root and receives the signal is pursued by the AMF mycelium, either AMF mycelium or the AMF spore, the MIC LCOs will be activated within the mycorrhizae, and this hypopodium will start established, and it allows the plant to recognition, you can say, MIC LCO. Okay, that is the main thing which helps in recognition of host, you can say, or you can say plant allows. So why there may be a question, lot of questions may be there, because the rhizosphere is considered to be the highly mobile area or active area wherein a lot of other pathogens will be there. Why can't the other pathogen gain entry? So that is the main thing. The plant defense will not allow. It is a selective one, you can say. Okay. So in this stage, you can see there are the establishment of these arbuscules, or how the arbuscules will form in a stepwise. In response, this is a pre-penetrating this apparatus. This penetration apparatus will be developed at the point of contact of the cell. You can see here PPA. Once this PPA is established, so the mycelium multiplies in that direction, and it forms a arbuscule-like structure within the cell. Okay. So this is the failed one, you can say. This is not the completely established one. This is the completely established one, mycorrhizae. So in order to distinguish between AMF and potential pathogen, as I mentioned, plants evolved a complex signal exchange with that of AMF, and this in turn, this AMF evolved signal pursue a suitable host. That is the main thing, as I mentioned. So this particular genetic signaling has been recorded right from this charophycean algae, and some of which are already in terrestrial in growth actually. So the charophyceae of algae, or charophyceae, whatever you pronounce. So that signaling component, what I mentioned, why the host plant allows only mycorrhizae and why can't the other pathogen? The main thing is a set of genes have been recognized, and the signaling cascade is there. So that is the main thing, wherein it plays a very important role in recognition, host recognition, you can say. Okay. Coming to this mycorrhizal role in sustainable nutrient mobilization and crop nutrition. Role of extended hyphal network in nutrient uptake. So as I mentioned, once the arbuscular mycorrhizae forms association, it sends mycelium extra root actually within the from the root, it sends outside the root, and it helps, that is the extended root colony, this mycelium will provide extra space for the root or the plant to search for the suitable nutrients. That is the main thing. And in case of ectomycorrhizae, it has been shown that the mycelial network is spread in an area of almost 2 kilometers. 2 kilometers. So this has been recorded, and I am not completely, I recall it's the plant name, but the ectomycorrhizal association, it has sent mycelia to the distance of 2 kilometers in search of nutrient. So that is the main reason the plant forms association, and that too particularly the larger surface area allows the better acquisition of phosphorus, okay, and other mineral nutrients, particularly micronutrients. So phosphorus is an immobile one, as you all know, immobile nutrient, and if though the phosphorus may be present 5 centimeter or 10 centimeter away from the root, the plant cannot access to it. But mycorrhizae can send the mycelium till that area, and there will be an accessibility, you can say. So that is the main thing. The plant forms association. So you can see here the external hyphae and the spores are germinated here. So the external hyphae will be occupying the space around the root. So this is the root. If you say the vicinity of the root, you can see. So it will be having the plant will be having an extended benefit of almost, you can see, say for example 10 centimeters or 20 centimeters or 30 centimeters till it reaches to the nutrient. Okay. So this is the plant uptake and mycorrhizal uptake pathway. Plant uptake pathway, you can see it is limited to this particular area. So though the phosphorus, nitrogen, if it is there just away from the root hairs, you can see within the, if it is not within the vicinity of the root hairs, it might be there away from the root hair. If it is there, the plant cannot access. Okay. But if mycorrhizal association is there within the plant, the mycorrhiza sends mycelium outside the plant. Okay. So though the root hair may be growing, say for example, till 5 centimeters, but it sends beyond that 5 centimeters and it reaches to the nutrient, and in turn, the plant will be get benefited, you can say. So that is the main purpose of this association, symbiotic association. So this is just another one picture, just I wanted to share. So in addition to that, mobilization of available nitrogen from the litter of the plants is also facilitated by AM extra-radical mycelium. Okay, and AM also improves to be the stronger candidate for low input sustainable agriculture, and through its active network of mycelium, it can help mobilizing nutrients from the crop residues itself. Okay, so this particularly if the person is following organic farming, you can say. So this mycorrhizae plays a very important role, and that can be considered as an organic farming also. Okay. So this is also another one, just to say that mycorrhizae plays in the interface between the plant and the soil in acquiring various nutrients. That is the main thing. And in turn, the mycorrhizae will get carbon source for its survival. Now the question is whether the AM ameliorates plant health through improved resistance to various abiotic and biotic stress. So that is about nutrient acquisition. Coming to this abiotic and biotic stress, yes, definitely mycorrhizae plays a very important role in overcoming abiotic and biotic stress. How? Salt affected soils makes about almost 10% of global land surface. I think day by day it is increasing as a result of continuous irrigation and accumulation of various salts, and that has that is leading to this salinity of the soil, and in turn to overcome this particular problem, mycorrhizae plays a very important role. Okay. And how exactly the phytotoxic effect of high salinity includes lower water potential in soil solution leading to decreased water uptake, then ion toxicity related to increased uptake of this chloride, Na and Na plus and Cl minus, and also reduced mineral uptake and absorption. So these are the side effects of, you can say, or the toxic effect of soil salinity. And the plants have a better sustenance against adverse effect of high salinity when colonized by AM fungi. How? Particularly when this mycorrhizal association it forms, it has got the mycorrhiza plays a very important role in release of, you can say, or in uptake of metabolites, particularly phytohormones, amino acids, vitamins, mineralizations, and solubilization process, providing nutrition and structural benefit to the plants. And production and accumulation of secondary metabolite, then osmotic adjustment under osmotic stress, then improved nitrogen fixation, enhanced photosynthetic rate, then increased resistance against biotic and abiotic stress. All these are the benefit the mycorrhizae can give. And in turn, in addition to that, mycorrhizae also helps in improving tolerance to heavy metals also. If the land is having heavy metal there, if the plant is accumulating a large amount of heavy metals that in turn will be toxic to human health, mycorrhizae reduces the accumulation of heavy metals by restricting the movement of heavy metals from the mycelium to within the plant. So that has been proved by conducting various experiments. Okay. So this slide shows the role of AM symbiosis in plant water stress. Particularly this physiological revamp, increase in stomatal density, then increase in water use efficiency, then stress-related gene expression, particularly aquaporin genes, ABA responsive genes, then dehydrin encoding genes, lipoxygenase genes, then hormonal balance, particularly decrease in ABA, because increase in ABA result in stress condition within the plant. So these mycorrhizal plants will help in decreasing ABA level. Then oxidative burst control also will be controlled by mycorrhizae by increase in SOD, then APX, GPX activities, then decrease in this H2O2 and MDA. Then osmotic adjustment, as I mentioned, solutes, inorganic and organic amino acids, vitamins, sugars, all these will be regulated. Okay. So these are the benefit in agriculture. Then mycorrhizal colonized plant may act as drought tolerance as well as drought avoiders, as most of the plants are able to adopt in a moisture deficient environment through two major strategies, that is drought avoidance and drought tolerance. Proline is considered to be one of the most widely distributed osmolyte, and the level of which that is particularly the abiotic stress, you can say, because this proline is considered to be one of the important protein which helps in overcoming the stress tolerance of abiotic stress particularly. So SOD, that is superoxide dismutate, is one of the most efficient antioxidant enzyme which initiates detoxification of free radical. Then ascorbate peroxidase is also another one enzyme abundantly found in chloroplast of mesophyll cells, and it's found to be carrying detoxification of H2O2 by absorbing glutathione cycle. Okay. And another one is glycol peroxidase is are the diverse group of enzymes which detoxify H2O2. improvement in soil moisture holding capacity of AMF, and AMF are known to produce a glycoprotein glomalin, and this glomalin is considered to be a glue. It acts as a glue-like structure forming aggregates of soil, actually aggregates of soil within the soil. So these aggregates of soil, the forming of aggregates of soil by this glomalin is considered to be a very important component in enhancing the architecture of the soil, and it also helps in retaining moisture. It has been proved. Okay, glomalin is considered to be one of the important contribution by mycorrhizae, and because of this, mycorrhizae can be considered as a best biological tool for improving soil structure. So, in addition to that, mycorrhizae also helps in plant hormone regulation, particularly during drought, as I mentioned. AMF have their positive influence on levels of jasmonate, terpenoids, carotenoids, and phenol. And this particular jasmonate is considered to be one of the important hormone which is synthesized within the plant in response to stress, particularly abiotic and biotic stress, you can say. Okay. So arbuscular mycorrhizae, a sustainable agriculture system, ecosystem system, sustainable ecosystem services. How? So plants in ecosystem performs a series of functions that are beneficial to well-being of human beings, providing multiple sources and processes, and trade-offs and links between plants and soil microbial communities can act as diverse of wide range of processes in ecosystem. What are these trade-offs? So mycorrhizal symbiosis is considered to be one of the fundamental play a fundamental role in providing ecosystem services due to beneficial functions of mycorrhizal fungi like plant fitness, then resilience against environmental stress, nutrient cycling, soil quality. Okay, so all these are the main thing. In addition to carbon fixation, actually, I think the much awaited research which is still going on, particularly how the microbial community helps in reducing the atmospheric carbon, because the microbes plays a very important role as a sink to carbon. So as the symbiotic, these are the symbiotic association formed between the plants and the green plants and mycorrhizae. The mycorrhizae requires carbon source from the host plant, okay, and in turn they supply nutrient. So these mycorrhizae can be a carbon sink. So that is what I mean to say is these mycorrhizae can be a carbon sink. So protection against pathogen is also one of the important benefits the plant can get if they form association with mycorrhizae. How? So the overall idea is before the pathogen enters the host, if mycorrhizae forms an association, it has been proved beyond doubt that mycorrhizae inhibits the entry of pathogen. That is one thing. Another one is phytohormones like salicylic acid and jasminic acid plays a crucial role in providing induced systemic resistance during pathogen invasion, and mycorrhizae have shown to enhance the level of these hormones significantly. So these two are the main component. And AM fungi also contribute towards improved uptake of nutrients and improved resistance to plants against soil-borne pathogen. So integrated response of AM colonization against pathogen. So activation of defense response. What is localized and induced resistance within the plant. Then priming of defense genes, particularly jasmonate and salicylic acid. Cell wall defense. Then defense-related protein production. Then defense-related enzyme activity. Then enzyme production of phenolic compounds and benzo oxynides. So these are the activation of defense response by mycorrhizae. And direct competition is also another one, as I mentioned. Competition for photosynthate, space, root space, then infection site and nutrients. So these are the competitive nature of mycorrhizae with that of pathogen. Then altered rhizosphere interaction, that is change in root exudation quality, then composition, then modulation of hormonal imbalance, influence of rhizosphere microbiome, then promotion to promote PGPR's population. So these are the contribution by mycorrhizae. And altered root morphology. So it has been proved that the enhanced lateral branching damage compensation will be done by if the plant forms an association with mycorrhizae. The enhanced nutrient acquisition, greater plant growth, increased tolerance to pathogen. So if the plant is healthy, then definitely it can have the ability to fight against pathogen. So that will be the benefit of mycorrhizae. So some of the papers just for which we have published, that influence of dual inoculation with mycorrhizal vesicular orbital mycorrhizae, particularly this Glomus aggregatum with that to Fusarium udum on plant growth and wilt disease incidents, which we have conducted experiment and we have proved that yes, mycorrhizae can have a positive influence in reducing the wilt disease of pigeon pea. Okay. And another one paper is effect of mycorrhizal fungi in managing the black bundle disease caused by Cephalosporin acrimonium. So coming to this plant growth promotion by AMF, as I mentioned in my earlier slide, that how exactly the plant will get benefited is because mycorrhizae increases this surface area for nutrient acquisition. Then it interact, then accommodating many soil organism through their hyphae. Okay, and because of all these things, mycorrhizae plays a very important role. So in to study, this is a paper just to mention which we have conducted, effect of arbuscular mycorrhizal fungi and growth and biomass enhancement of Piper longum, and we found that definitely the mycorrhizal inoculated plant has better growth when compared to non-mycorrhizal plants. So this experiment we have conducted, and we have used 3 species of Glomus. That is one species of Glomus, another one species of Acalospora, and another one Gigaspora. 3 different genus of mycorrhizae. With Central Asiatic also, we found that the mycorrhizal plant performed well when compared to non-mycorrhizal plant. And with sugarcane also, we found, you can see there is a remarkable difference with mycorrhizae and without mycorrhizae. There is a clear demarcation, or in growth particularly, growth enhancement and the vigor of the plant if you want to see. Okay. With Vigna radiata also, we have conducted our experiment and proved that mycorrhizal plants are better when compared to non-mycorrhizal plants. And this is also the same thing. So So this nutrient uptake, biocontrol and eco-restoration, if you want to see. So even at the low levels, you can see the plant performed well. Low level of elements or nutrients, plant performs well when the mycorrhizae is there in that vicinity. Okay, that is the thing. Another one is, as I mentioned, that heavy metals will be filtered. They act as a filters. Mycorrhizae act as a filters if the plant is is growing in a heavy metal contaminated area. How exactly? So whatever the heavy metal which is there will be accumulated within the plants, within the mycorrhizae, you can say, and only the nutrient will be supplied. So the mycorrhizae acts as a filters for heavy metal acquisition. So if in the absence of this mycorrhizae, you can see directly the root might absorb actually, and it accumulates within the host plant, and the plant may become contaminated with the heavy metals. I think this you can skip. This is the old one. So coming to this, whether the mycorrhizae can be mass produced. Of course, I think there are technologies as of now they have developed that in vitro cultivation can be done, in vitro culturing can be done, but it is very difficult because it requires sort of technical expert. But through trap culture, a single pure culture can be multiplied. If you isolate the mycorrhizae, identify it, and if you inoculate on the host plant by trap culturing method, you can multiply it, and the whole of the root mass which is produced can be used as an inoculum. So that is the main thing which we have conducted in our laboratory, and we have mass produced. So that is the main thing just I wanted to share with you. So I think this we can skip. What are the feature perspective? So mycorrhizal research, in future, it will be, you can conduct experiments in studying the sustainable agriculture by using mycorrhizal cultures, then plant growth and productivity field, the mineral nutrition, water uptake, then managing rate of photosynthesis, accumulation of secondary metabolites or active ingredients. So these are few things. Then abiotic, environmental stress tolerance, disease resistance, soil health and fertility, ecosystem functioning and biodiversity, reclamation of degraded land, then bioremediation, then preventing soil erosion through this production of glomalin, as I mentioned. So these are the few future perspective one can look for. And to conclude, arbuscular mycorrhizal fungi can be used as biofertilizer to enhance the ecosystem functioning and agricultural productivity without disturbing the balance of ecosystem and environment. That is the first main thing just I want to mention, because continuously if you add synthetic fertilizers, we are losing a lot of biodiversity, particularly microbial diversity. And arbuscular mycorrhizal fungi as characteristics, they make them to potential tool to be utilized in a sustainable management of agriculture and environment, you can say. Okay. And therefore, the field of inoculation with mycorrhizae, depending upon the type of plant species and their environmental condition, could be an effective alternative to agrochemicals. So that is the main conclusion just I want to make. Okay. So if you have any doubts, you can ask me. Whatever I know, I can clarify. Otherwise, we can share.
Yes, sir. Yes, sir. Yes, sir. I am Somlingappa. I have an sir. Actually, I know that some species of the plants, that is Acaranthus aspera and the Parthenium, associated always with the mycorrhizal fungus. Because of this reason, the Parthenium found occur everywhere. It grows as a weed. That is the weed in the agricultural land.
Yes, sir.
It is impossible for us to control. Even it grows in anywhere else in the topsoil, very fertile soil, and in the marshy places as well.
Yes, sir.
Both these two plants. Other than these two plants, I think Eupatorium.
Eupatorium, yes.
Yeah, Eupatorium is also becoming, and already it has become one of the very, that very strong weed.
Yes, sir.
It is impossible for us to control that is the multiplication, or in other words, the growth and development of the Eupatorium. Recently, it is also observed that this Eupatorium is also growing along with the cultivating plants, cultivating in the field, in the farmer's field, sir.
Yes, sir.
My question is that Eupatorium plant also associated with this the mycorrhizal fungus, sir?
Yes, sir. I think almost all plants, they are having a mycorrhizal association. Only few species, I think 80 to 90% of the angiosperms. Eupatorium is also mycorrhizal in nature. But mycorrhizal association and the weed occupancy of the land, actually, it doesn't have any correlation actually, because the production of speed at which Parthenium produces and dissemination of seeds, seedling particle. That is the main reason why it occupies the main this one, and the speed at which it occupies, okay, the empty land is the main reason. It is not, it cannot correlate between the mycorrhizal association and the spread of weed, you can say. There is no correlation.
Yes, sir. Other than this Eupatorium and then the Parthenium plant, even one can observe that is the mass of, that is a very wide number of plants on the all along the roadside. That is a Cassia pistula, sir. What it is called? Congress. Then sorry, BJP, sir. BJP, sir. Actually, in this part it is called as a BJP, sir. Cassia pistula.
This is a new term for me, BJP Kasa.
Okay. Parthenium is called Congress Kasa, sir.
No, there is a reason behind that actually. That's why it is called Congress. But BJP, I was not knowing.
Yes, in this part, actually, Kashyap Pistula.
Sir, I have one question. Sir, I am Modi, sir.
Yes, sir. Yes, sir. Please, sir.
Sir, Modi, sir, Gulbarga.
Yeah, tell me, sir. Modi, sir. Tell me.
I want to ask one application part of mycorrhizae, sir. I have some desired plants where I want to association of mycorrhizae. So what extent we have to do, sir?
So, if you want to grow a plant and if you want to make association of mycorrhizae, normally in natural condition, if you are growing, then definitely mycorrhizae will be there in the soil. If the plant is, if the land is having pre-grown plants there. So if you want to sterilize it, sterilize the soil, and if you want to add mycorrhizae, then you can select a good mycorrhizae and you can add it, and automatically if you sow the seed, the plant forms association. There is no doubt.
Is there any enemies for the mycorrhizae, sir, for the stoppage of luxuriant growth and association, sir? For
mycorrhizae?
Yes, sir.
I think mycorrhizae, there is no enemy. But the only thing is, if it has to find a suitable host actually. Until and unless it gets into host, it will not proliferate. Sir, because being obligate in nature, it will not proliferate. That is the main thing.
Sir, actually, how it finds the host, whether the chemotactic attraction or something?
Yes. As I mentioned, strigolactone. Strigolactone is a hormone released into the as a root exudate.
Yes, sir.
Root exudate, it releases, the host releases a hormone, strigolactone. So in response to that strigolactone, the mycorrhizal spore germinates and it multiplies towards that particular the exudate.
So it means we have to introduce that strigolactone in all the angiospermic plant, desired angiospermic plant.
So
is it possible, sir?
Yeah, if you want more pollination, then you can in the rhizosporic zone you can add this strigolactone. Otherwise, naturally the plant tend to produce strigolactone actually.
Yes, sir.
Yes, sir.
Thank you, sir.
Yes, sir. You're welcome. Good afternoon, sir. Tell me.
Good afternoon, sir.
Yeah.
Sir, how to identify these? in the soil there are many fungi are there, AM fungi. So how to identify and how to culture this?
So identification part, as I mentioned, identification is mostly if you want to go for morphological identification, then you have to look for its spore wall, spore the color of the spore walls, and hyphal attachment, and type of hyphal attachment, how exactly the hyphal nature is there. So these are the characteristic. Then the content within the spore. So these are the characteristic features we can look for identifying morphological base. If you want to go for, as of now, I think molecular characterization is also possible. For which you need to have the single spore culture. You have to go for single spore culture. Once you get a sufficient spore, you have to extract DNA, then you have to use a suitable primers. Then you can go for molecular characterization. Two aspects are there, sir. But normally people will go for morphological identification. Yes.
Yeah, preliminarily morphological identification, then we can go for molecular.
Yes, sir. Yes, sir. Yes, sir. Morphologically, I think glomus species, you can differentiate glomus, then gigaspora and aclospora broadly.
Yes, sir.
Within the glomus species, if you want to identify at the subspecies level, it is a bit difficult. You have to accurately record the characteristic feature. But broadly, between the genus, that is glomus, gigaspora, aclospora, sclerocystis, you can clearly differentiate actually.
Yes, sir. Definitely, I am interested in this area, sir, because I did a little work on this mycorrhizae.
Okay.
So I want to work with this.
Yeah, definitely. We can identify it. I can identify it. No problem. We can.
Yeah, thank you, sir. Thank you, sir.
Yes, sir. Yes, sir. Yes, sir.
Sir, arbuscular mycorrhizal fungal spores are available in large scale for the farmers to supply for the supply of the farmers or to.
Sir, can you repeat the question? Arbuscular mycorrhizal supply.
Arbuscular mycorrhizal fungal spores are available in the market in a large scale.
Yes, sir.
Which are available to our farmers to use in their field, sir.
Yes, sir. I think commercially they will sell in different brand names. It is available. Biofertilizer, it is there. Availability is there, sir.
Under the name the biofertilizers.
Yeah, biofertilizer. Yes.
Available in the market, sir.
Yes, sir. Yes, sir. It is there.
Okay. Thank you, sir.
Yes, sir.
If there are any questions,
sir?
I'm asking, sir, my fellow participants.
Okay.
Questions are there.
Okay,
so
already.
I want to give word of thanks.
Okay, sir.
So if there are no questions, I would like to thank Professor Rajkumar, Department of Studies in Botany, University of Mysore, for giving elaborative information on mycorrhizae and the different aspects, how it is useful, and their importance in the soil, how they are important to for the soil fertility. I thank you so much, sir, on behalf of myself, on behalf of HRDC, UGC-HRDC, University of Mysore, and also on behalf of all my fellow participants. I thank you very much, sir.
Thank you, sir. Thank you for patience hearing, and we can collaborate. Actually, anybody, if you have any query, you can always send me, and even I can identify it. No problem.
Yeah, thank you, sir. Thank you.
Yes, sir. Yes, sir. All the best. Wish you all the best.
Thank you so much.
Thank you, sir. Thank you, sir.
Thank you, sir.
Shall I close it? Or you will be online only?
Yes, sir. You can,
No, sir.
You will be online. Okay. Thank you. Wish you all the best. Yes.
Thank you.
Thank you, sir.
Thank you. All the best, Modi.
Thank you, sir. Thank you.
Palled also.
Thank you very much, sir. In future, coming days, I will contact you, sir.
Please come. Bye.
Yes, sir. Yes, sir. Thank you, sir.
Sir, sir, sign only.